This video will cover question 68 from the biology section of the 2018 NSAA paper. In this question, we're told that we have a plasmid which has been made recombinant by the insertion of a plant gene for herbicide resistance. So here's our diagram, and it's labelled with different shaded regions. Our gene for herbicide resistance, which is what's made this plasmid recombinant, i.e. a new piece of DNA has been introduced, is labelled in red. We also have a green region which codes for resistance to tetracycline, an antibiotic, and we have a region coding for resistance to penicillin, another antibiotic. So this is the recombinant plasmid, and we're told that bacteria containing only the recombinant plasmid were separated from bacteria containing the original plasmid. We're also told, as we can see in the diagram, that the bacteria contain genes coding for resistance to these two antibiotics, and that they were then grown on two agar plates. One of the plates had penicillin, and the other had tetracycline on it. We're then given an answer table, and we're asked to identify the row which shows the growth of these two cultures on the two sets of agars. So here are our column headings, and I'll summarise the contents of the table. So here are our column headings, and we have two rows per answer row, each row relating to either the recombinant or the original plasmid. The recombinant plasmid, remember, has this extra gene for herbicide resistance, which is in the middle of this gene for the antibiotic tetracycline. So we can think that this gene interrupting the gene coding for resistance will make it non-functional, and therefore the bacterium that's recombinant will now be susceptible to the antibiotic tetracycline. So when it comes to penicillin, since both maintain their resistance gene, it's not been interrupted, we'll have, in the case of the recombinant plasmid, yes, it will be resistant to penicillin, but it will not be resistant to tetracycline because that gene has been interrupted. The original plasmid will be resistant to both penicillin and to tetracycline because the gene has not been interrupted by this herbicide resistance gene. So we have our answer that we have a row representing this information, and that answer is going to be option C.